Before I start, I'd like to thank the organizers for the opportunity. So how did our species evolve to be who we are has been a question of long-standing interest. How did we become humans? We tried to answer that question by looking for functional elements that were gained in the anthropoid primates and asked, could they explain the unique primate traits that we see or any trends in human evolution? Before I go any further, what are anthropoids? Anthropoids are primates that are closer to humans, so that would be apes and monkeys. So why look for functional elements specific to anthropoids if you're studying human evolution? True that most studies in human evolution have focused on this branch, events that happen after the split from the chimpanzee, but not all of human evolution occurred here. Most of our human specialized traits are also traits that are found in primates as well. For example, the increased brain size that you see in humans was just a culmination of an event that started in the earliest primates. So it is imperative to look at this longer branch where most of our primate evolution happened. So that's what we did. We looked at this longer branch, and why do we do that? The point that I'm saying here is that if we understand how we became primates, we get a better handle on human biology and evolution. Not just that, there's a biomedical relevance as well. Understanding how our genome differs from the commonly used mammalian model systems could help us define limitations of the model organisms that we normally use, the rodents, and perhaps even design better models. Thanks to the Marmoset Consortium, now we have the Marmoset Genome Assembly available, and so that you can use this to answer questions about the descent of anthropoids from mammal ancestors. And why is that possible? Because Marmoset, being a New World monkey, it shares biology and features with the humans. At the same time, it lies on a distant branch of the anthropoid tree, thereby providing sufficient statistical power to detect constraint. Note that I'm detecting constraint here because constraint is indicative of function. So what kind of sequences am I looking at? These are the ones. Anthropoid-specific constraint elements would look like this. So I'm looking for sequences that are highly constrained within the anthropoid primates, whereas the orthologous mammal sequences would be evolving at neutral rates. So how do we do this? The strategy would be to first find anthropoid constraints sequences or elements, and then throw away those elements which are also constrained within the mammals. Right? So Rick, my colleague who handles the computational aspects of the study, put together a custom bioinformatic pipeline to do this. So first, he aligned the genomes of these anthropoid species. He did a whole genome alignment and threw away those with bad quality alignments. He was left with clean syntenic alignments. And he ran GUMBI, which is a constraint dissection algorithm. He used a very strict threshold, and we got 268,000 anthropoid constraint elements, which I will be referring to as ACs henceforth. So here we had our anthropoid constraint elements. Now we have to subtract mammalian constraint from this. And so for that, we had to find that set. So we wanted to detect even any trace of faint mammalian constraints. So we looked into 4 different subgroups of mammals, used 2 different constraint algorithms, combined the elements detected by them. And most importantly, we used very generous thresholds so that even weak constraint could be detected. And as you can see, the red circle is much bigger than the blue. We identified almost 2.1 million non-primate mammal constrained elements. So now you intersect them, and what you're left with would be anthropoid-specific constrained elements. And we had 23,849 of them. So this represents the most dramatic primate-specific elements that one could ever find in the human genome. So here we had our sets of ASCs, and the next obvious question was, are they false positives of our algorithm? So to test that, if they were false positives, they would not correlate with any measure of functionality, and so we used the derived allele frequency distribution as a measure of test. So if you look at the black bars, they represent the SNPs in the African population. SNPs that are deleterious or cause variation occur less commonly, and we all know that. So in other words, they have lower derived allele frequency. If you look at the SNPs within ASCs, they show even more downward shift. So that indicates that SNPs within ASCs are rare, or if they do, they have an effect on the population. So this kind of nicely gives us an evidence that ASCs are functional in the human population. As an additional test, we use the ENCODE's DNase hypersensitivity data across 84 human cell lines, and we asked, are our ASCs enriched for hypersensitive sites? In other words, are they enriched for regulatory functions? So what you see here is that right at the center of ASC, you see, I keep saying ASC. ASC is an anthropoid-specific element that we identified. So right at the center of ASC, you can find a 2.3-fold enrichment for hypersensitive regions, saying that these elements are likely to be in open chromatin regions than random. So we were nicely convinced that they have an evidence of functionality. So the next obvious question was, what are their properties? Especially, how much of them lay in the coding and in the non-coding regions. So you see here that the anthropoid constraint sets, the ACs, 13% of them lie in the coding regions. In stark contrast, if you look at the anthropoid-specific elements, only 0.3% of them are in the coding. So that's a 47-fold reduction. Well, overwhelming non-coding bias of this may not surprise many of us, right? But 99.7%, this magnitude is quite unexpected, and it provides a strong support for Mary Claire King's hypothesis that primate evolution primarily occurred through cis-regulatory changes. So we knew that they could be functional. We knew that they had an overwhelming non-coding bias. The next question was, are they enriched for any functional categories, especially for functional categories that could explain primate evolution? So we used the GREAT algorithm and looked for enriched annotations in the genes flanking ASCs. So I've got to mention that we used ACs, the anthropoid constraint set, as our background set and looked for relative enrichment in ASCs. So for one time, I'm not going to show you the full results, but I'll highlight a few major themes that we came across. We saw an excess of enrichment for multiple neurotransmitter receptors like heterotrimeric G protein receptors, serotonergic and GABAergic receptors. And what you see on the right is the FDRQ values for that. Well, we were wondering what primate trait does it correlate with. We found this interesting paper from Pasco-Rakish's group that talks about overexpression of a bunch of neurotransmitter receptors in the primate brain, especially in the face when the when our cerebral cortex expands. So this nicely correlated with the enrichment that we saw for ASCs, and we hypothesized that ASCs near these neurotransmitter genes could have in some way contributed to the primate-specific regulation. Well, we all know that primates have good visual acuity, and this is partly because of our lens thickness. We saw excess enrichment of ASCs in the lens genes and the genes that are involved in the lens development, quite coherent to the known primate traits. When we looked into the human phenotype ontology, most of the terms that came up were motor phenotypes like clumsiness, microcephaly, and gait ataxia. Our group is very interested in finding out if these ASCs could have in any way contributed to our unique motor skills and perhaps even the primate-specific aspects of our motor diseases. So in line with that, I'd like to show you the top 20 genes genome-wide that were enriched for ASCs. And if you look at it, the bottom line is that they are genes that are implicated in anxiety and panic disorders, in Parkinson's, in a couple of motor disorders, in autism, and cognitive disorders as well. Well, the interesting thing from here is that some of these disorders have primate-specific symptoms, and they are hard to model in rodents, right? People generally don't report failed model systems, but we found an interesting paper which talked about the UB3A knockout model in mice for Angelman syndrome. Angelman syndrome slightly borders on mental retardation and motor phenotypes, and they said that the mouse model did not replicate two main cardinal traits that you see in human patients. So this failure of the mouse model, they said that it's because, of the many reasons. One reason was because UB3A is differentially regulated in humans and in primates. I'm sorry, did I say primates? Humans and in mice. Right? So in order to solve this issue, they said, what about cloning human genomic regions into the mouse model and humanizing the mouse model? And thereby, you could address this issue. So this is where our ASCs would come in handy. Right? If you are looking for candidates where you can humanize any transgenic mouse model that has failed, then ASCs are the best place to start. Enough talking, and what about experimental validation of the ASCs that we have? So before we went on to do that, we said, do they have any in vivo functions? So we used the roadmap epigenomics data and looked at the enrichment for ASCs across 8 human fetal tissues for the data that's available. And what you see here is that ASCs are excessively enriched in the fetal brain, quite unexpected because brain regulation is complex and conserved. But compared to the ASCs, there's a significant enrichment in ASC, which is pretty interesting as well. And the second strongest signal was the fetal placenta. So this immediately brings to mind 2 major primate traits, right? Altered primate brain regulation and specialized placenta to support our extended gestation. This is pretty interesting. So we picked one of our candidates from here. We said we are going to test it for enhancer activity in developing mouse embryos. So we did the mouse transgenic enhancer assay, and what you see here is the result of one of the candidates that we tested. So human ASC 192, the anthropoid-specific element, drove expression, was an enhancer at 11.5, and it drove expression in the brain, in the eye, and in the spinal cord. Okay. So our hypothesis here is that ASCs were non-functional in the mammals, and they gain functionality in the primates. So to test this, we had to assay the mouse and the dog orthologs. So that's exact, or any mammal orthologs. We chose the mouse and the dog ortholog. And what you see here is that the mouse ortholog fails to be an enhancer. There's no strong and reproducible pattern of expression. Neither does the dog ortholog. I'm showing you just 4 representative embryos here. We had a couple more. It flows to 13 to 15. So to get more clues about this enhancer, we looked at higher resolution, and what you see here is the spatial pattern of this enhancer. So this enhancer drives expression in the ventricular regions of the brain and in the neuroepithelium. So what does this mean? These are the sites from which new neurons arise. These are the sites where neurogenesis happens, right? So we were interested, and before we would further pursue this, we wanted to ask which is the target gene of this enhancer. So we did a 3C assay on human ES cells because human fetal tissue at day 30 is quite unavailable. So the question we asked here was that which promoter does this enhancer regulate? And we found that there was long-range interactions of the enhancer with the 2 promoters of POW2F1, whereas it does not interact with the promoter of PREG1 or CD247, the other genes. This is pretty interesting because POW2F1 is commonly known as OCT1. Maybe many of you would know that. It's a developmental transcription factor which controls the growth of radial glia cells. So what are radial glia cells? They are the stem cells that generate new neurons, right? So if you tie the spatial pattern of ASC 192 and the function of OCT1, they both point to one thing, neurogenesis. So it's our hypothesis that ASC 192 is doing something during the neurogenesis phase of the primate brain. We are continuously knock-in and knock-out studies of this enhancer are in the pipeline, so I'm not going to show you that. So the summary, to summarize, we identified a set of constraint elements, the first genome-wide signals for primate evolution. They have an overwhelming non-coding bias, and they are the molecular correlates of unique primate traits. So we know that the primates had these traits, but what was the molecular basis of these traits? ASCs could be interesting candidates to test. So do they have a role in primate-specific disease traits? This remains to be tested. We've identified a first primate-specific developmental enhancer. So to end, I'd like to thank Shyam, my PI, for his enthusiasm and support, Rick, my colleague, the rest of the CSME members, A staff for funding, and of course, Kim Wally for having us part of the Marmoset Consortium.
Thank you for a nice presentation. My question is, experiments, when a human-specific element is put into a mouse, then you actually do see expression that recapitulates the human expression, suggesting that the trans, like more.
Exactly.
In your case, you're actually finding something that expression does not happen. So could you comment on what your interpretation is about the trans?
Well, if you test the human element, we do see expression pattern in the developing mouse. The first panel that I showed was the human element tested in the mouse.
I see, and then you take the
mouse.
mouse.
The top 2 were the ortholog. I mean, the ones that were below, the second and the third panel, were the mouse and the dog ortholog.
So your interpretation is still that the evolution is still driven by cis changes, while the trans is more.
Exactly. We might miss out candidates that are different because they have a different set of transcription factors. But right here, we are more focusing on cis regulation.
Thank you.
Great talk. I always love seeing the innovative work coming out of Singapore. I did my postdoc there as well years ago. I didn't catch the point about the DNA's one sites. Are they more enriched in the ASCs than mammalian wide set, or you didn't actually do that comparison?
Oh, we did that comparison, and ASCs You're talking about the human cell lines or the fetal tissue?
No, I'm talking about the, so wherever you took the DNase 1 hypersensitive site data, so you showed ASC data, but is that more enriched compared to like all mammalian-wide elements, or it's less or equally enriched?
Yes, we did throw in FastCon elements as well, and our ACs to test how they were comparative to that. Actually, FastCon elements and ACs are better, way higher, a little way higher. In fact, it's similar to FastCons. Our ASCs, our Anthropoid-Specific elements, are similar to FastCon elements, but ACs are way more higher, and that's because most of them are coding, and in coding you see extreme constraint, right? So, yeah.
Right, and how many of these ASCs are directly transcribed? For example, or encode RNA-seq data?
RNAs, around 4.5, yeah. Yeah, not much. Only a similar fraction of ASCs and ASCs intersect non-coding RNA.
You mean 4.5% of the ASCs?
I'm sorry.
Or?
0.45, yeah, 0.45. 0.45 percentage, yes.
Let's thank.
I can take it offline.
Let's thank Nirmala once again.